Hello, in this video we are going to go over the concept of DNA replication. We're going to be learning how cells make a copy of their DNA. Remember that DNA is the main molecule that cells use to store their genetic information. So in this diagram, this is just a simple diagram of DNA. Hopefully you remember that DNA is in a helix shape and it has two strands. So it's made up of two strands of nucleotides. And the order of the nucleotides is how the information is stored. And so as you can see here, we've got A's, T's, G's, and C's in a very specific order, and that specific order is how the cell stores its information. Kind of like how we store information using letters. These letters in specific orders mean something to the cell. Now, because of how precious DNA is, it carries all of the information that the cell needs. It is going to be very important that when it makes a copy of its DNA, that copy does not have any mistakes. And so in this video, we're going to be learning about how a cell makes a copy of its DNA and avoids, obviously, making any mistakes. So before we get started on how DNA replication actually happens, I first wanted to go over the structure of DNA in a little bit more detail. So DNA, as I mentioned, one molecule of DNA has two strands, so one strand on one side and another strand on the other side. And these strands actually have a direction. And the way that we name the direction is based on the carbons. So when we're looking at a nucleotide, remember that nucleotides are made up of a phosphate, a sugar, and a base. Now that sugar is a deoxyribose sugar if we're talking about DNA. And that deoxyribose sugar has 5 carbons, which we number them: 1', 2', 3', 4', and 5'. So the little asterisk means prime in this case, but essentially we're just numbering the carbons. Now, carbon number 5 is important because that's where the phosphate group is attached to, and carbon number 3 is important because that's where the OH group is attached to. So 5 and 3 are basically opposites of each other. Carbon number 5 is on one end, and carbon number 3 is on the other end of a nucleotide. And so during DNA replication, it's really important for us to keep track of the direction because nucleotides can only be added to the 3' end. So I like to think of it as the 5' end is like the back end, and the 3' end is the front end. And we can only add new nucleotides to that OH group on the 3' end. We can't add nucleotides to the 5' end. And so most of the time in biology, we designate that DNA has a direction, and that is a 5 to 3 direction. So we always write it in the 5' to 3' direction, and that is, again, because during DNA replication, that is how nucleotides are added. Now, another thing to note about DNA is that it is anti-parallel, or also known as bidirectional. So one strand of nucleotides goes 5' to 3' in one direction, and then the other strand of nucleotides on the other side goes in the other direction. So very similar to how cars on the road go. On one side of the road, you're going in this direction. On the other side of the road, you're going in the opposite direction. And so DNA is similar to a road where we have two directions, one on one side, and then the other side goes in the opposite direction. And this is going to be really important when we talk about how DNA gets replicated, which is why I wanted to talk about it here. So now let's go over the process of DNA replication. By the way, in this video we are simplifying it a lot. I will have another video where I go over it in more detail, but this is kind of the introduction to DNA replication. So let's first look at our original piece of DNA. This is just a random piece of DNA, and notice that I have labeled it 5' to 3' and then 5 to 3 on the other side. Remember that DNA is bidirectional, so one side goes one way and the other side goes the other way. So the first step in DNA replication is this enzyme called helicase is going to separate the two strands. So DNA, remember, is double-stranded. We're going to separate those two strands. And again, we're keeping track of where the 5 to 3 directions are, but for now it doesn't really matter. So that's the first step. Now, once these strands are completely separated, then RNA primers are added. So another enzyme called primase is going to add something called the RNA primer. Now, what is a primer? If you've ever painted or used makeup, primer is the thing that you add before you actually put your makeup or your paint, whatever it is. And so in DNA replication, before we actually add our DNA, we have to add RNA. It's going to seem silly, but you'll see in a second. But this is just how it works. So primase, which is an enzyme, comes and adds these RNA nucleotides to make an RNA primer, and it's going to be adding it in the 5 to 3 direction. So meaning it's going to start at the 5' end of the strand that it's making. So it's going to be on the 3' end of the original strand, but the 5' end of the new strand that's being made. And it's going to go in that direction because that's the 5 to 3 direction. And on the other strand, same thing. There's another RNA primer being made by primase. So usually there's 2 primases that are working at the same time. So the primase over here is adding it in the 5 to 3 direction, going that way. And it is adding RNA nucleotides, which seems weird because we are making DNA, but you'll see in a second that those are just going to be removed. And I've designated them as kind of this orange-yellow color just to distinguish them. So these are RNA molecules being added. Now the primer is not very long, anywhere between 5 to 15 nucleotides. It's just that first piece that we have to put. Okay, so step 3 is really where the DNA is being made. And so DNA polymerase, so a totally different enzyme called DNA polymerase, is going to come, and it's going to basically continue. So it starts where the primer ends, and it's going to continue, but now it's going to add DNA nucleotides. And guess what? It's still going to go in the 5 to 3 direction, so we're starting at the 5' end of the new strand, and it's going to continue adding nucleotides going in the 5 to 3 direction. So for this DNA polymerase, it's going that way. For this DNA polymerase, it's going that way. Now it's just going to keep going along the DNA until it's done. And our final result, sorry, I'm blocking it a little bit. Our final result is that we're going to have 2 identical pieces of DNA, and each molecule of DNA is going to have 1 old strand, which I've designated with the blue color, and 1 new strand, which I've designated with the red/orange color. And so we call this the semi-conservative way of replicating DNA because guess what? Half of the DNA is from the original old DNA, and then half of the molecule is now new DNA. Now you're probably wondering, what about those RNA molecules from the primer? Well, basically another polymerase comes in, removes the RNA primer, and replaces it with DNA. So that seems ridiculous, so this whole step is just going to get removed and replaced, but that's just how life works. So, these enzymes aren't perfect, and this is just basically how evolution designed it, or I guess not designed, but you know what I mean, just it is what it is. So primase has to come first, then DNA polymerase, and then a different DNA polymerase removes the primer and replaces it with more DNA. So seems like an extra step, and it kind of is, but that is just how life works. So that is kind of DNA replication in a nutshell. And those are kind of the main steps. Now, there's definitely other enzymes involved. In this diagram, I'm only talking about the main 3. We're talking about helicase, primase, and DNA polymerase. But in another video, I'll go over the kind of nitty-gritty, smaller details of DNA replication, and there's many more enzymes that are involved. But if you're looking at this diagram as a whole, this is pretty much the main steps that happen in DNA replication. Now, the last thing I wanted to do before I ended this video was talk about how the polymerase even knows how to add the nucleotides. Which nucleotides does it add? Remember that DNA has information, and so it doesn't just add nucleotides randomly. It has to add the correct nucleotides. So how does it know which nucleotides to add? It's going to be using the base pairing rules. So DNA polymerase, and also RNA primase, by the way, any kind of molecule that is adding nucleotides to create DNA or RNA, the way it works is it uses the base pairing rules. So as a reminder, A pairs with T, or if we're talking about RNA, that's going to be U, and then C pairs with G. So what I wanted to do in this diagram is kind of try it out. So let's act like we are DNA polymerase. So here is our original strand of DNA, and the strands have been separated by helicase. So let's just pretend that we are DNA polymerase, and we are going to be adding new nucleotides in. So remember that we need to follow the 5' to 3' 5' to 3' direction. So I can't start here, and the reason why is because if I go this way, this is going to be this 3' end of the new DNA, and this is going to be the 5' end of the new DNA. Remember that DNA has to go in the opposite direction. So if this is the 5' end, the other side has to be the 3' end. So that means I actually have to go backwards this way. Now if I'm DNA polymerase and I see a letter T, I know for a fact that a letter A is going to go here, because A and T are the only two that pair. And then I just keep going in this direction. So G pairs with C, A pairs with T, another T, a C, another C, A, C, T, C, A, and T. And that is what DNA polymerase is essentially doing, is it's looking at these nucleotides that are unpaired and is adding the correct pair. Now, if I were DNA polymerase and I was on the bottom strand, then I would start here, because this is the 5' end for this new this new strand, and we're going to be going in this direction. Let me fix that. And then this is going to be the 3' end. So we're going in this direction now, because this strand is going in the opposite. And same thing, we're going to add an A because there's a T there, and we're going to add a T because there's an A there, and we're just going to kind of keep going in that direction. So G, A, G, T, G, G, A, G, and T. And that's pretty much it. So DNA polymerase just kind of follows along, going in the 5' to 3' direction, and it's looking at the other strand and basically making sure that it matches it by following the base pairing rules. And if you look, hopefully I did it right. This strand, in terms of the letters, should be identical to this strand, and they should both be identical to the original strand. And I'm just going to double check real quick. Looks good. And that is essentially how DNA is replicated in terms of the actual molecules, how the information is being copied over. So hopefully this video was helpful, and I will see you in the next video.